Hello and welcome to this Australian Biocommons webinar. My name is Melissa Burke. I'm the Australian Biocommons Training Manager and I'll be your host for today. This webinar is the first in a series focusing on leveraging deep learning to design custom protein binding proteins. The series has been developed by the Australian Structural Biology Computing community and is designed to inspire and help you recognise the potential applications of these new approaches. Before we begin, I would like to pause and take a moment to acknowledge the traditional owners and their custodianship of the lands on which we meet today. We pay our respects to their ancestors and their descendants who continue cultural and spiritual connections to country, and we recognise their valuable contributions to Australian and global society. Today, I'm joining you from Brisbane, and I acknowledge the Turrbal and Yagara people as the traditional owners of the land on which I am joining you from. Today, we are thrilled to welcome Dr. Rhys Grinter from the University of Melbourne to the webinar to speak to us about using AI protein design to design binding proteins to challenging bacterial transporters. Rhys is the Grimwade Research Fellow and runs the Molecular Physiology of Microbial Pathogens Lab in the Department of Biochemistry and Pharmacology at the University of Melbourne. He has a PhD from the University of Glasgow, and in 2021, he founded his lab and was awarded an NHMRC investigator grant to continue his work on ion piracy in bacterial pathogens. He uses current and emerging technologies to understand how microbes work, to prevent disease, and to develop enzymes and natural products as tools for biotechnology. He uses diverse techniques across genomics, molecular microbiology, biochemistry, and structural biology. Welcome to the webinar, Rhys. I'll hand it over to you.
Awesome. Thanks so much. Thanks for the really kind introduction, Melissa, and to you and the Biocommons team for putting together these webinars. It's a really nice platform to discuss some recent advances in this space. And also thanks to Kate Mitchie as well for so much effort bringing the Australian structural biology community together for these to discuss these new computational methods. So today I'm going to discuss some recent work that we've just had published using de novo protein binders to inhibit heme uptake in pathogenic E. coli, and use that as a platform to more generally discuss how to implement this technology and also where I think it's going and where the potential is. I'm going to go through some of the basic science first, partially because I'm excited about it because it was just published, but also because I think it sets the stage for the kind of background information you need to really leverage these technologies successfully. So yeah, without further ado, I'm Rhys Grinter, obviously, by the way, and I'm at the University of Melbourne. Okay, so this work, we were lucky enough to have this work published just last week in Nature Communications. And so if you want to have a detailed read of the paper, have a look here. The experimental work was led by Daniel Fox, who's finishing his PhD in my lab, and it was a team effort between my lab and the lab of Associate Professor Gavin Knott at Monash University. And obviously these days to do one of these kind of papers, it takes a village, so a whole bunch of other people contributed significantly as well. So the heme piracy I'm talking about, the ion piracy I'm talking about is it's performed by these TONB dependent transporters. And so TONB dependent transporters are a family of proteins that I've been looking at for quite a while. They're produced by gram-negative bacteria and they exist in their outer membrane. So if you remember your microbiology 101, you'll probably remember that gram-negative bacteria have an outer membrane and an inner membrane, and they have a periplasmic space in the middle. And these TONB-dependent transporters, they sit in the outer membrane and they capture rare and precious substrates for the bacteria. So usually ion-containing compounds, but other stuff as well. And they solve a really unique problem in that the outer membrane of gram-negative bacteria, it's separated from the rest of the cell and it doesn't have any inherent energy source. So these things are 22-stranded beta barrels that are plugged by an internal plug domain. And basically, for the ligand to be imported, this plug domain needs to be displaced. And this is performed by an inner membrane complex called the TONB-EXBB-D complex. And that's powered by the proton motor force in the inner membrane and has this kind of long arm that grabs the transporter, displaces the plug domain and gets the ligand inside the cell. And I think when in my PhD, I just thought this was a really cool mechanism and so started studying these transporters then. but they're also really important for obtaining essential nutrients during pathogenesis for pathogenic gram-negative bacteria. And one of the most pathogenic gram-negative bacteria, serious pathogens, is E. coli, pathogenic E. coli. It's the main cause of urinary tract infections, and it's a significant cause of gastrointestinal infections. And when I say significant, I mean hundreds of millions of cases a year and hundreds of thousands of deaths. So it's really a big problem, especially in the developing world. And CHU-A, which is the Tom-B-dependent transporter I'm going to talk about today, is produced by pathogenic E. coli, and it imports the iron-containing cofactor heme. And heme is the most abundant source of iron in the body. And preliminary work on CHU-A before we started this study shows that CHU-A can target heme directly, or it can target heme in association with the protein called hemoglobin, which is in our blood. And so heme is a super important nutrient source for bacteria. And so in certain infection contexts, CHU-A is an important virulence factor during systemic infection. So this gives us the why this system is important to investigate and try and target. So studying iron uptake is kind of fun, but it's also very difficult in gram-negative bacteria because iron is such an important nutrient that the uptake systems are incredibly redundant. So even in your lab E. coli strain, there's 7 or 8 Tom-B-dependent transporters are involved in iron uptake, and they're all pretty redundant in function. And so you can't really study the phenotype of one without the other ones getting in the way. So quite a few years ago now, published in 2019, I developed an E. coli strain where I deleted all the native iron uptake transporters and got a really nice clean phenotype that you can see here when you grow this strain under increasingly iron-limiting conditions by adding bipyridine to the agar. It won't grow. And so this gives you a really clean background for investigating the function of iron-containing proteins. And so to leverage this system, I took the operon that contains this Tom-B-dependent transporter CHU-A and other proteins involved in heme uptake, and through recombination, I introduced the genes into the E. coli, into the genome of this E. coli knockout strain, with the idea that it would allow E. coli to grow on hemoglobin as an iron source, and then you could knock out this transporter and do mutagenesis and manipulate the system in that way. And that worked quite well. This is a hemoglobin-containing agar plate that's otherwise iron-limited, and the knockout strain doesn't grow, but when you have the CHU operon, it grows quite well. And then when you knock out CHU-A, it doesn't grow, and you can complement this in a plasmid-based system. So that shows the importance of this transporter for getting heme from the environment. And so this is where Daniel Fox, really talented PhD student in my lab, joined the project. And so Daniel took this strain and really wanted to drill down and characterize what were the preferred substrates for CHU-A, because there was sort of not exactly conflicting information in the literature, but it was sort of known that it could target free heme or hemoglobin or myoglobin, but its specificity in what its physiological substrate was less clear. And so Daniel initially did agar growth assays with the different heme sources in the agar, and basically showed that yes, CHU-A can be used to import heme from hemoglobin as an iron source. It can import heme or myoglobin. And then he did a series of really detailed liquid growth assays to show that CHU-A has by far the highest affinity for hemoglobin, either alpha beta hemoglobin, which is ordinary hemoglobin, or alpha gamma hemoglobin, which is fetal hemoglobin. And it had a lower affinity for heme and a much lower affinity for myoglobin, and wasn't able to target heme-containing proteins like neuroglobin, cytoglobin, or the iron sulfur cluster-containing protein ferredoxin. And so this sort of suggested to us that hemoglobin is likely the physiological substrate for this transporter, considering also that the majority of heme in our bodies is bound to hemoglobin. Daniel also did some BLI experiments to show the affinity of CHU-A for hemoglobin, and showed that it has a pretty high affinity. It's around 80 nanomolar. However, it has quite a fast on rate and off rate. And this was consistent with what we'd previously seen, what other people had seen in the literature, where there's basically a mechanism where CHU-A is able to bind hemoglobin rapidly. It extracts the heme out of hemoglobin and then disassociates from the complex. And so this sort of mechanism kind of miraculously to me occurs without any external input of energy, happens freely in solution, but as a really kind of intricate catalytic, extraction mechanism. And so we wanted to determine the experimental structure of CHU-A bound to hemoglobin. And so basically set up some crystal trays where we mixed hemoglobin and CHU-A, got some nice pink crystals and solved the structure, but only ended up with this map here, which is basically CHU-A by itself with heme bound. And so this is the post heme extraction state from hemoglobin. So we got this structure, which was informative, but not quite what we wanted. And then Daniel followed up on this by doing cryo-EM on 2A with hemoglobin, but we only ever saw class averages that it was 2A by itself, which was further indicating that you've got this rapid binding, heme extraction, and dissociation, which made it very difficult to capture this complex experimentally. The other thing I kind of wanted to just note for context as well is that probably, as everyone knows, hemoglobin, when it's inside our red blood cells, is predominantly a heterotetramer, so it's 2 alpha subunits and 2 beta subunits, but actually exists as an equilibrium of a tetramer and a dimer state. And this is both redox state dependent, the redox state of the heme, whether it's +2 or +3, and pH dependent. And the literature is actually pretty hazy in solution, at least, what are the triggers that cause this equilibrium to shift. And so we kind of struggled for a while to sort of test the different forms. And so Daniel did some really nice systematic analysis showing that reduced hemoglobin, oxyhemoglobin, is basically primarily tetrameric at pH 8, but as soon as you lower the pH to pH 6, it becomes basically dimeric. And with methemoglobin, which is the oxidized form, this relationship also occurs. And so this, plus information from the literature, and the fact that when hemoglobin is bound by another protein, haptoglobin, in the serum, it's a dimer, sort of indicates that the dimer state is the most likely state that the pathogen is going to experience. So using this information, we performed Alphafold modeling on 2A in complex with hemoglobin. And so predictably, the tetramer didn't seem to give us a reasonable solution. But for the dimer, the alpha beta dimer of hemoglobin, we got this structure where the beta subunit is interacting with loops 7 and 8 of 2A, and then the alpha subunit is kind of hanging out into space. And I'll just show a little video of that. So this is alpha beta hemoglobin in complex with 2A, Alphafold model. And then this is alpha gamma hemoglobin with a gamma subunit in pink here in complex with 2A as well. And you can see the binding modes of those are the same, as is the binding mode of myoglobin, which also has the globin fold, but is monomeric. And so the Alphafold confidence scores for these models were quite reasonable for the alpha beta hemoglobin in complex with 2A, and a little worse for alpha gamma and for myoglobin. However, considering they all had the same binding mode, we thought there was a pretty strong indication that this could be the physiological representative of the physiological complex. Another thing that kind of gave us a clue that this was probably right was when, if you look at where heme is in the hemoglobin, alpha beta hemoglobin, alpha gamma hemoglobin, and myoglobin complexes, they're all in a similar location. And when you contrast where that heme is in the bound hemoglobin predicted complex to the heme we saw in our crystal structure, you can see that the coordination of heme in the crystal structure is very close to where the heme is in the hemoglobin. And you could see how conformational change could cause release and delivery of that heme to that histidine. So we think that gave us some quite reasonable insight to the structure, to the model of binding and extraction that we couldn't get from an experimental model. And so to sort of confirm this, we want to do some hypothesis-driven research, because we had a couple of complexes of hemoglobin in complex with a nanobody. And also we could form a complex with this serum protein, haptoglobin, which scavenges free hemoglobin, so it's not available to pathogens and it's not in the serum. And so we looked at the structures of these complexes in comparison to our Alphafold model. And we saw with the nanobody that the nanobody binding site was nowhere near the interaction interface with 2A, and so hypothesized that shouldn't affect uptake. And then you've got your haptoglobin in pink in the middle in complex with your hemoglobin. And when you superimpose that complex, you're getting some quite bad clashes with those static structures. And so we hypothesized that that would probably affect the ability of 2A to access the heme from hemoglobin. And so Daniel tested this in a agar assay, where you basically have ion-limiting agar, which you overlay with your bacteria, and then you spot on the purified protein complexes at zero dilution, and you see what your lowest growth concentration is. And so we saw for hemoglobin by itself and hemoglobin in complex with a nanobody, we got the same level of growth stimulation, indicating that they're equally good at providing heme to the bacteria. And then with the alpha beta hemoglobin in complex with haptoglobin, we saw a marked decrease in the ability to access this substrate, which indicates it's a last substrate. And so, a worse substrate. And so this broadly fits with our structural data. We kind of expected, based on the structural model, that the hemoglobin haptoglobin complex would be completely inaccessible to the transporter. But I think that neglects the fact that these transporters have quite a lot of flexibility, and there's also the possibility for flexibility in that hemoglobin haptoglobin complex. So we think that that model is probably not entirely correct and that there's flexibility. But it still provides a strong indication that our Alphafold model is at least reasonably correct. And the final thing I wanted to talk about in terms of mechanism before we go on and talk about binder design is heme binding. And so there was information in the literature showing that in CHU-A and some other transporters, two histidines play a key role in heme binding and import. And that's histidine 420 here, which is the one we saw bound to heme in our crystal structure, and then histidine 86 here. And so we were doing some work with Alphafold 3 because that allows the modeling of ligands. And we got these 5 different solutions from our Alphafold 3 run that have different coordination states for this heme molecule. And one of them matches our crystal structure with heme just coordinated by histidine 420. And then the other states move towards a bidentate coordination of the histidine between histidine 420 and histidine 86. And I thought this was quite telling, considering the known role of histidine 86 in the function of this transporter. And that probably represents some conformational dynamics that occur in solution or occur upon transport of CHU-A. And this broadly fitted with some spectroscopic data that Daniel performed. And so don't worry too much about the peak heights here. But basically, when you incubate CHU-A with hemoglobin, you get heme transfer to CHU-A and you see a peak representative of absorption of heme at 413 nanometers. And when you mutate either histidine 420 or histidine 86, you see a shift in the wavelength of that peak. And that's indicative of a change in the chemical environment of the heme. And that happens when either histidine is mutated, which indicates that they're both playing a role in coordinating the heme in the complex. Whereas you don't see that when this histidine 68 is mutated, which is over here, distant in the inside of the barrel, where the height of the peak stays the wavelength of the peak stays the same, basically. And so that provides a strong indication that these histidines are also important for heme binding and import. So to put this all together as context for our de novo modeling, we kind of basically established a model where you have a dimer of hemoglobin binding to loops 7 and 8 of CHU-A. And then you have dynamic changes which causes extraction of the heme and binding to histidine 420 and probably histidine 86. And then you have energization by that TOM-B-EXBB-D complex, which causes import into the cell, and that's how the bacteria get their heme. And so about the time that we were kind of finishing off this work, some new tools for AI protein design came out, and we thought this would be an interesting system to kind of explore with some potential therapeutic applications. And so we used RFdiffusion, which I would guess probably at this stage most people in the audience are familiar with. The preprint was released in 2022, and the paper came out in 2023 from David Baker's lab, Nobel Prize winning lab. And this was kind of it was a real leap in the ability to do de novo protein design and de novo binder design, because it had a much higher success rate. It produced backbones with a much higher success rate than previous methods. And so this method leverages the Rosetta Fold structural prediction program, and basically uses a denoising algorithm, trains a denoising algorithm to understand how to make realistic protein backbones, starting from some prompts and from Gaussian noise. This only produced polyglycine backbones, so it doesn't produce any sequence. But luckily, the Baker lab had also been working on this, and they designed a graph neural network called ProteinMPNN, which is very good at looking at a given protein structure and then providing a sequence which will cause that protein to fold and be highly stable and expressible. And so if you combine these two things together with Alphafold 2 for structural prediction and validation, you can basically create a pipeline for protein design and also binder design. And so for binder design, what you do is you provide a target model as a static full atom representation. And then you basically can tell RFdiffusion, I want you to hallucinate a backbone to a various region defined by amino acid hotspots on this protein. And then RFdiffusion will do various iterations, as many as basically you tell it to, making different backbones starting from random noise. And you can change the noising settings to get more or less diversity. And this is kind of how this works here. You basically start out with a random collection of atoms of your length, and then basically it diffuses a protein or protein backbone that looks like this. And so our strategy for design was to take what we knew about the CHU-A hemoglobin complex. Excuse the different colors here, but this is CHU-A. And this is our Alphafold model of the dimer of hemoglobin bound to CHU-A. And we were like, if we make binders that target these two loops, bind to these two loops that hemoglobin interacts with in our model, we should be able to block hemoglobin binding and then iron uptake by the bacteria. And that was the overall hypothesis. And I guess I would just reinforce here that you need to have this biological understanding to really target biological systems. And so the nuts and bolts of our binder design campaign for this project were to use RFdiffusion for backbone design, using QA as the target model, doing sequence generation using ProteinMPNN, and then filtering and validation using Alphafold 2. Specifically, implementation of ProteinMPNN and Alphafold 2 called DL binder design, which uses a stripped down version of Alphafold 2, which is very fast. We generated 20,000 designs in silico, and then we screened them using Alphafold 2 to get designs with a confidence, a PAE interaction confidence score of less than 10 and a PLDT score of greater than 90. And from this, we shortlisted around 400 binders and then performed manual curation to get 96 for synthesis for screening. I'm not going to go into too much detail about the manual curation, but we looked at things like delta G of binding, surface area, folds we like the look of, and fold diversity. It's unclear which of these are effective or most effective, and part of it was just we had to take our 400 designs which worked and then turn them into 96. So yeah, I think a lot of these things do have potential, but I don't want to too strongly advocate for any kind of additional selection criteria. The binders we designed were between 70 and 120 amino acids in length. We selected 96 of them, and then we assigned a gene sequence to them, a DNA sequence for expression in E. coli using the Twist Biosciences web portal, and then got Twist to synthesize the 96 genes with his tags in expression plasmids as clonal genes. So we made the decision here to go with clonal genes, even though it was a bit more expensive, because the effort and time cost of cloning all of these proteins and all these genes, and the things that can go wrong, I think make it a bit preclude the efficiency of that route. And this way we get NGS verified clonal genes from Twist, which we can put straight into an expression E. coli strain, and with a really quick turnaround. And so for this project, which we did in 2024, we ordered the genes on the 16th of February and we received them on the 7th of March and could go straight into screening and testing. And I think for a lot of applications for binder design, this extremely rapid approach, especially stuff like pandemic response, this extremely rapid turnaround and having rapid pipelines is really important. So you have the DNA in your hand, transform it into a E. coli expression strain, and we did this in a 96-well plate, and we did recovery and then antibiotic selection directly in the well. So no plating out to plates and selecting individual colonies, just using the antibiotic selection to kill any of the bacteria that didn't have the plasmid. And we found this works quite well. We tested it with fluorescent protein because we were a little bit skeptical, and we got robust expression of GFP in all of the wells routinely when we did this. And so we don't think that plating out is necessary, and this massively simplifies the workflow. Then transferred these expression strains to autoinduction media in 24-well plates and grew the cells overnight to express the protein, harvested the plates by centrifugation, did chemical lysis in the 24-well plate format before adding nickel resin to bind the protein. And then we used a vacuum manifold in either a 24-well or 96-well format to rinse, to wash the resin and then elute the protein. I'll just get a little video of this device here. And so this is it's a pretty simple apparatus. It's from Cytiva, so obviously it wasn't cheap, but it's hugely enabling because it allows you to do all of that chromatography in parallel and very quickly go from, your cell lysates into 96 purified proteins in a plate. And so doing this, we could basically go from DNA to protein for screening in 3 days, which when you combine that with the DNA synthesis time gives you a little under 3 weeks for doing this whole process. And with a good rinsing regime, we achieved quite good purities, definitely sufficient purity for the next steps. And so then after all that work, it was time to see if these things actually worked. And this was before there were that many reports in the literature of this technique working. And, I have a lot of faith in the Baker lab, but we were very nervous that this 7-month kind of design, screening, scrambling campaign to implement this technology, we were very nervous whether our designs were going to work and our assays were going to work. And so basically to test this, we set up a phenotypic assay where we had a plate that had hemoglobin in it and then we overlaid our Tomb-dependent transporter CHUA strain on that plate. So it should be able to grow using the hemoglobin as an iron source, and then basically spotted on each of our purified binders onto the plate to see if we got any zones of growth inhibition, because if the binders had a high enough affinity, they should be able to compete with CHUA for access to the transporter, and then they should basically block growth. And after setting this experiment up on a Friday, Daniel set it up, and there was a very nervous weekend of waiting. But then basically came in on the Monday and had a really nice result. It's sort of the result that I was kind of imagining and actually dreaming about in my head, stress dreaming, because we saw some really nice zones of inhibition where some of the binders had been able to inhibit growth by blocking access to hemoglobin. And across the board, it looked like a lot of the binders had some activity, but then there were a few key standouts. Probably about 4 or 5% of the binders had high affinity, or they were effective at blocking hemoglobin uptake, which is about what they saw in Bindcraft within the range of success. So that was fantastic. We basically picked the winners from this process. The other thing that was interesting was if you put heme in the plate instead of hemoglobin, so just free heme, the growth wasn't affected by the binders. So CHUA is still able to access free heme, even when the binder is presumably interacting with the transporter. And we think that goes down to the mechanism of binding of those two histidines that I'll discuss a little bit later. So then we picked the winners, A10, C8, G7, and H3. And Daniel did a lot of really intensive characterization to figure out the level of growth inhibition on agar, which was down to a low nanomolar level. Here's the Alphafold models of the designs in complex with CHUA here. And so you can see they're all alpha helical bundles, which is quite common with RFdiffusion, but with different topologies and completely different sequences. And so they're completely unique proteins, completely unique entities that are having the same effect based on their design parameters. Daniel next assessed the ability to inhibit growth in liquid culture to get an IC50, and basically showed that we got from a low micromolar to a relatively low nanomolar IC50, showing these things are quite potent at blocking hemoglobin uptake, even when hemoglobin was present at 100 nanomolars, so at a higher concentration than what some of the binders were, which was quite gratifying to see. He then followed this up with a lot of BLI to look at the affinities of these binders for the transporter. And we got a range of affinities in the kind of tens to low hundreds of nanomolar range for a dissociation constant. One thing that I thought was quite interesting was that A10 we measured to have an affinity of 155 nanomolar, which was comparable to G7 and H3. But when you look at the growth inhibition assay, A10 was only able to inhibit down to about 3 micromolar as opposed to these to 50 or 100 nanomolar. And our hypothesis is that comes down to the fact that A10 has a much higher on and off rate, as you can see from the BLI, which potentially means that it's through dissociation, it gives hemoglobin the chance to interact with the transporter and then for heme extraction to occur, because that's quite rapid. And so we think for this application, having a slower on or off rate is probably going to be quite important for efficacy of these binders. But the big question that we had was, these binders are clearly doing what we designed them to do, but do they structurally match what we designed, and how closely do they match? And so initially for that, we turned to X-ray crystallography. We set up crystal trays for all of the binders, and predictably for crystallography, we managed to get crystals for one of them, which we solved by X-ray diffraction, and basically showed that yes, the individual binder by itself in solution very closely matched the computational design. So the computational design is in pink, experimental structure is in red, and the RMSD for most of the atoms was about 0.55 angstroms. And this was comparable to the variation between the different molecules in the asymmetric unit of the crystal. So there were 8 different molecules in the asymmetric unit. And so we think within kind of flexibility and experimental error, the computational design basically matches the binder, the experimental structure for the binder, which was really nice to see. But that was one thing, but do these things look the same in complex with the transporter? And so to do that, Daniel turned to cryo-EM with the help of Brad Spicer, Chris Lupton and Harry at the Monash cryo-EM facility, and managed to prepare grids and collect data for ApoCHUA, CHUA with G7 bound and CHUA with H3 bound. And one of the things I really like about doing cryo-EM is if you're doing an experiment like this, you can see whether you've got a result or not as soon as you get the class averages, because you can see that in both of these, the transporters now have a little hat, and so the binder is present. And so we solved that structure and basically managed to show that if you compare these structures to our crystal structure with a heme bound, that you've clearly got very clear density for the binders bound at the location, interacting with loops 7 and 8 as we predicted, or as we designed, basically. And this was a very close match for what the design should have been. If you look at the overlay between the binders alone, the RMSD was around 0.63 angstroms for most of the residues. And then when you look at the whole complex, clearly the complexes match very closely. There's some misalignment of the binders, but when you analyze the structure, you can see that misalignment is driven actually by the flexibility of loops 7 and 8, and not so much by a difference in the fold of the binders. When you align them by themselves, they're very close. And this was more pronounced for binder H3, but you can see absolutely it's driven by a change in conformation of these loops. And when you overlay just the binder here, they're actually very close, and probably the differences are driven more by flexibility than poor prediction. And so this was kind of a really nice proof of concept for us, and I think for the community, that these tools are really robust for creating binders which work, but also creating binders that exquisitely match the design from a structural perspective. And this allows us to leverage them to really modulate a lot of different systems. And this is one of the things we're really excited about. And so just to sum up the kind of the biology, the science design part of this talk, here's our kind of model for how all this is working. So you've got a 2A in complex with the binder, and you can see when you overlay that Alphafold model of the alpha beta hemoglobin complex, you've got a really large surface area overlap between these two molecules, which should preclude binding. But when you look at heme bound by itself to 420 and the position of that histidine 86, it's not very occluded, and that's probably why heme is still an effective substrate for the transporter with these binders bound. And you also see that for H3 as a binder. Big overlap of the binding interface and where the heme would get extracted based on our model. But with heme by itself, these two histidines are still relatively free to engage in binding. And so we don't think this would be probably an issue for the application of these things as therapeutics, because the heme that bacteria are likely to see in the body is very mumuch going to be bound to proteins, it's very much going to be bound to hemoglobin, but we could do further design to target heme inhibition as well. So what's next with this project? Well, of course, pathogenic E. coli has a second heme uptake transporter, which is relevant HMA. So we're now working on designs of binders to target that as well, kind of as a dual therapeutic. We've put an ideas grant in to maybe get some money to do this. And so hopefully that gets up and we can do some work on it. And then if that does work, we'll design the binders and optimize them, and then start testing them in infection models of uropathogenic E. coli in mice. And that'll give us an indication of how well these things work. We know that these transporters are important for infection and infection in this model. And it's also going to provide some really valuable data on immunogenicity and stability and what's the best design parameters for using these things as therapeutics, which I think is something which really needs to be addressed to move forward. And so that's it for the structural component of my talk. It looks like I've still got another 10 or so minutes. So I just wanted to kind of I was thinking about this yesterday and sort of go through kind of at a high level my thoughts of what's required if you want to set this up and do it yourself and you haven't done it and you haven't really thought about it too much. And these slides are quite text heavy, and I apologize for that, but I was thinking, considering it's a webinar, people can revisit them and get an idea of what they might need to do. And I've sort of done this into 3 stages. Stage 0, which is setting up the compute, stage 1, which is implementing the pipeline, and stage 2, which is doing the testing of the designs. And so stage 0, you're going to need access to HPC compute, which is going to allow you to run these programs. It's possible to run them on stuff like Google Colab online, but the compute we've found is probably going to be insufficient except for the easiest targets for a generation campaign. And so I would kind of advise sustained access to between 2 and 8 GPUs with at least 24 gigabytes of VRAM as a requirement for doing sustained binding. And unless you want to completely rewrite all the code, you're going to need NVIDIA GPUs. You can see here, this is a lab serve we bought for doing this work in 2024. We got two 4090 GPUs, 56 threads, about 1/4 terabyte of RAM and a bunch of hard drive space. And this set us back about 20K from a commercial retailer. So expensive, but not out of control considering what lab equipment costs. And this is sufficient for doing kind of a standard binder design campaign if you didn't have access to a university HPC cluster. So we use a combination of this lab server and university clusters, so Spartan at Melbourne, which is quite good. And we generally use the lab server for design and scoping and some binder generation. And then if we want to do a full campaign, we'll lean on the HPC compute for access to more GPUs. One thing I'd say is don't be afraid of consumer grade GPU hardware. It's much cheaper. It's louder and hotter and more power hungry, but it's much cheaper. But just be aware that the VRAM will limit you for some applications. So RFdiffusion and ProteinMPNN are basically fine with 24 gigabytes, but Bindcraft with larger models, it's going to take longer and it's not going to run as well without more GPU RAM. There are national pipelines in construction, as far as I'm aware, using servers like Galaxy. So they're an option, and you can also use cloud computing, although I think this probably requires a bit more understanding of the underlying architecture of the programs to do. And then you've got to pick the programs that you're going to use to do this. There's really an explosion in this space of new algorithms and new implementations and new strategies for protein design and binder design. We've kind of limited ourselves to those that have been published and rigorously experimentally tested in the study, because otherwise there's a risk that it's not going to work in reality as it does on the computer. And basically, today I'll talk about RFdiffusion, which I discussed before, and Bindcraft, which is a more recent binder design pipeline. There's also proprietary binder design programs like Alphafold from DeepMind, which is not publicly available, although you can request access to a relationship, and Chai2, which was very recently released, but is not yet available to the public. So I already discussed RFdiffusion, ProteinMPNN, so I won't go into that into too much detail. But last year, Bindcraft was developed by Martin Persea in Bruno Carrera's lab. And we've used this for a number of projects now, and when it works, it does a really good job. Experimental success rates are much higher than RFdiffusion, which means you can make less models. It's open source, there's no commercial restrictions. And the only drawback I would say is it has a higher GPU VRAM requirement, and it gets quite slow if your system is more than, say, 400 amino acids. So definitely worth giving this a try, and it's pretty simple to implement. Alphafold is another option. It wasn't benchmarked against Bindcraft, but it has been tested against RFdiffusion. It works quite well. However, it's not publicly available, and it's got reported success rates of up to 88%. And Chai Discovery, that I think was released just a couple of weeks ago, it's also made by a company, so it's not publicly available as of yet, but it has a high success rate for de novo binders, and they're also reporting a significant success rate for de novo antibodies, so around 16% against 50% of the targets they tried, which, if we start to have access to tools like that, you're going to be able to design, antibodies in a 24-well plate from the computer, which I think is going to be a real game changer for a lot of applications. So you've decided what programs you're going to want to use. So let's say RFdiffusion and Bindcraft, and you want to install them on a cluster or a lab server. This is going to require some experience or some upskilling with Linux, Bash, Python, and Conda. But I would emphasize you don't need to be an expert. So the binders that we made for QA, I had very little experience with these tools and kind of taught myself how to do it over the period of a couple of months. The process will be frustrating, at least initially. Any open source software has inherently got bugs and contradictions between libraries, and Conda's a bit temperamental, I would say. But a few tips that I would say if you want to start out. There's a company called Lambda Stack, which makes computers, but also provides an automatic tool for updating and installing AI software packages like PyTorch and making sure your CUDA and versions all are compatible. And so this can save a lot of headaches getting all your machine learning packages onto your Linux machine. Reviewing GitHub pages carefully are very helpful, especially Bindcraft and RFdiffusion have quite good instructions for how to run and install the programs, and the developers will engage in helping you troubleshoot. ChatGPT I found hugely enabling, both for understanding, how to use these tools, and also for generating some really simple code and scripts. And for someone who's a lab head and is quite time poor, this was really what bridged the gap to allow me to be able to do this without having to spend the time to learn how to code in Python really well. And tenacity in troubleshooting, reaching out to developers and community for advice. If you've experienced a bug, someone else has probably experienced it. And I'd say while there's a lot of benefit for developing sophisticated pipelines and automating all of this stuff, to get started, you don't need to have a sophisticated pipeline to get good results. All of the results we did in that Nature Communications paper, they were all from ChatGPT generated Python scripts that we cobbled together and run on whatever hardware we had available. So if you're interested, I would say, and you want to put in the effort, get started, and don't let perfect be the enemy of good, I guess would be my perspective. So now how am I going for time? I got a couple of minutes. I can finish off. So you've got your pipeline installed, and so now you need a target basically. And so I would choose an experimental structure or a computational model. We've had quite good success with some computational models, but make sure that it's high quality. So less than 2.5 angstroms resolution and with good B factors is probably a good rule of thumb for experimental structures, and high confidence scores for computational models in the region you're modeling on. So if you're using Alphafold 2, great PLDT score of greater than 90 for the residues you're looking at. You trim your model to the smallest possible size you can make a binder to, because compute costs scale at roughly a square scale with residue count. And so if you have a big model, it's going to take a long time to get your designs and you're going to struggle to get critical mass. And also beware of what I like to call binder escape. So if you over trim your target and you leave a hydrophobic core exposed, the binding program will ignore your hotspots and then just make binders to the hydrophobic core, and you'll get good scores and you'll get your hopes up, but it won't work. So you've got to kind of have this balance between the size of your model being as small as it can, but not allowing the binder program to cheat and not make stuff that you want. And basically then select your target, trim your target, make a number of, figure out where you want to make the binders to and identify hotspot residues. And that'll tell your binding program where to make the binder to. I can't go into the details of this now, but all this documentation is on GitHub pages and in papers. And then normally we would do some scoping runs, so small scale runs of RFdiffusion and Bindcraft to figure out parameters where we're getting binders that pass in silico screening metrics, and also we like the look of from visual inspection. And this can sometimes be very easy with some targets and can be difficult with others, and can be incredibly hard with some targets using the current tools. It can be very difficult for some sites to actually get any binders that work. And to get around that, you can change hotspot location, change binder generation model settings and models, and get creative with your design strategies, or even reach out to the developers and try and collaborate to get some more cutting edge tools are all options. Basically, but once you've found binders you like the look of and they're doing what you want them to do, then you can scale up. And so this probably should go a bit earlier, and I've kind of discussed it, but it's very important to understand the structural and biological context of what you're making a binder to, so it actually works. But once you have a set of parameters that you like, then you can do a larger scale binder generation campaign. And for RFdiffusion, the RFdiffusion based pipeline, we generally would make 200 to 400 binders that pass in silico success rates, and that can require making anything from 1,000 to maybe 20 or 30,000 models in total. And for Bindcraft, we would generally make 50 to 100 designs. So the difference in how Bindcraft and RFdiffusion works is that RFdiffusion makes all the models and then you've got to screen them yourself, whereas Bindcraft iteratively screens for quality during the design process and will only give you the models that pass that. So basically everything that comes out of it is kind of good to go in a way. And this will probably generally it'll take you days to maybe a month of GPU time. And so if you've a month of GPU time split over five or six GPUs is still only about a few days. And then curate your binders. We select for fold diversity, interaction interface, binding energy, the kind of fold, and then just create a set of binders that we like, considering all the things that we can consider, and then synthesize those.
But then once you've finalized the designs that you want to test, you want to screen in the wet lab, we basically would select between 96 binders for RFdiffusion and 24 to 48 binders for Bindcraft, and that's because of the high relative success rate of Bindcraft binders. And then we would send the DNA sequences that we derive from the protein sequences that were designed to Twist Biosciences to synthesize as clonal genes. And this is generally costing between $70 and $100 per binder. And this is still a significant expense, right? Because, if you've got 48 binders, it's going to be about $3,500. If you've got 96, it's going to be probably around $7,000. But the rapid turnaround, the fact they're NGS verified, and the fact that they're in clonal expression vectors ready to go when you get them, I think kind of makes this cost justified. It's possible to order gene blocks, and for some applications, you could use the gene blocks directly. But if you want to do expression in bacterial cells, cloning them yourself isn't really cost effective when you account for people's time, and also the fact that you're probably not going to get 96 clones happening all at once at the first time, and so you'll have to, repeat some of the cloning, and it'll kind of mess with the workflow and the time. And so we really like this idea of getting the DNA synthesized rapidly and then rapidly getting it into the lab so we can test it. And so, yeah, I've sort of talked about how we did the small scale expression before in the content of my talk, so I won't go through this in too much detail, but we generally achieve 80 to 90% success rate with binder expression. And from a small scale expression on 4 mils or so of culture, we usually get between 100 and 500 micrograms of binder roughly. And this is more than enough, in its 96 well plate format for identifying which binders are good to go on with. Just that there. And so then once we have our binders made, we'll generally have them in a 96 well plate. And then we perform assays to figure out which ones are working, because the success rates are still, the majority of your binders still probably aren't going to work very well. And so our philosophy here is to use whatever assay gives the fastest result, whether that's a biochemical assay, biophysical assay, or a cell-based biological assay. And the aim here is to identify several good binders, and only having semi-quantitative or qualitative data for this, I think, is okay. And one of the tools that we really like for this is BLI, biolayer interferometry, because this is kind of a good compromise between throughput and the level of quantification it gives you. So if for this, you need to have your target protein kind of purified effectively, and then you kind of attach it to a chip and then use interference to see if your binder's binding. And here's an example of what one of those binder campaigns might look like. So this is, I think, 48 Bindcraft designs that we've screened against the target. And we're basically just looking for binders where we see a response, and then, I guess the slowest off rate possible if we're looking for a high affinity binder. And so from this data, you interrogate your data and then pick the binders that you really like the look of, and then move forward with those for further analysis. You can also use like a cell-based assay, for example, and cross-correlation between two methods is great, but not essential. And and so if you're going to use a cell-based assay, you could do something like this here, where you're not really probably going to have a lot of certainty over the concentration of your binder in the assay, and so you're not going to be able to determine exact, association, dissociation, affinity, but you will get a kind of a relative impression, for example, of how these binders are performing compared to the native ligand, which is quite nice. But yeah, this is not the only way of screening binders, basically. So depending on your experimental setup and your target, you could do things like a pooled library of DNA for binders, which you're selecting in a cell-based assay, or intracellular expression of discrete binders if you've got an intracellular target, or even in vitro translation combined with an enzyme assay with a purified enzyme. So it's sort of up to you, and your imagination is what's sort of the imagination and your experimental system. It will dictate, the kind of assay you can come up with to test your binders. But fast and quick, I think, is kind of the key. And so then once you've identified your winners from that initial screen, we would select a few binders for scale up. So we generally do maybe 4 or 5 binders and produce them in milligram quantities, either in a coli or potentially in another cell system, depending on what you want. And then rigorously quantify them. So quantify binder affinity, on rates, off rates, modulation of activity of the target, and then potentially structural characterization of the complex. And so this is kind of an interesting one, because there's a lot of literature and work that we've done that basically shows that this the experimental structure is more or less identical to the model. And so it kind of begs the question of whether or not you actually need to do this, expensive and time-consuming structural characterization to prove that the thing is real. And I think over time, as we kind of get more used to this technology and trust that these models are in fact correct, if they're working in the way that we would think they would work, I'm not sure this structural characterization is actually going to be necessary. And you can do further affinity maturation on your binders if you don't quite achieve the affinity you want. And this could be done experimentally through something like phage display. So then you're effectively using your AI part as your initial stage to get a binder and to make sure you're selecting the right target, but then you're boosting the affinity experimentally. Or you can also use computational methods like partial diffusion, which basically add some noise into the model of your binder bound to its target, and then allow the binder to kind of diffuse out, allow the program to diffuse out that noise so you get variation on the binder scaffold in its sequence. And so this can also boost the affinity. And so once you've done that and you're happy with your binder, then in order to kind of translate it into whatever use you need, you will need to do things like assessing its stability. And if you're going to use it in a sort of in a therapeutic sense, it's assessing its toxicity in mammalian cells and immunogenicity. And this is sort of the stage that we're kind of at now, where we've got binders and we're starting to move them through this pipeline so that they can be translated into things that can be used in the real world. So here's kind of just the sort of summary of the workflow that I've got sort of discussed here. Basically, you have the target, you can have an experimental structure or a structural model, you identify where you want your binder to bind to, you use the AI tools to generate the binder, and then once you've done your selection, design campaign, worked out the ones you want to test in the lab, we use a synthetic DNA provider, so we use Twist to synthesize the DNA, get that into lab, reproduce the binders recombinantly on a small scale, screen them, and then upscale and rigorously quantify their properties so they can be used as therapeutics, diagnostics, or research tools. That's it for the experimental part of the talk. And so I just kind of wanted to, in the last couple of minutes, go through some of my thoughts about the future challenges for AI-guided protein design and where some of the opportunities might be. And so one of the things that's still a limitation with the current generation of software is some target sites and some proteins are much easier to target than others. And this could be because of something like, a protein might be embedded in the membrane, so it's very difficult to access that transmembrane region in a chemically sensible way. But also surfaces like polar surfaces and really flat surfaces are still pretty difficult to make binders to. And I expect that this will improve over time, but it does mean sometimes extensive scoping or kind of more creative design strategies are required to target certain targets. Explicitly accounting for post-translational modifications is also still difficult. So stuff like, glycosylation or, lipidation and stuff isn't really well, or ligands isn't really well parameterized in the prediction and the generation models. And so at the moment, we're kind of just avoiding post-translational modification sites, but as the tools for, prediction of the structures of these chemical entities improves, I expect that this will be integrated. So you should be able to explicitly have a protein which binds to a glycosyl site and to the protein. There's still more work should be done in this space. Experimental success rates could always be better. We achieved about a 4% success rate with nanomolar affinity binders for 2A, which is okay, but it still necessitates synthesizing, say, around 96 constructs, which means it's a significant expense in terms of getting the DNA synthesized. The cheaper it is, if you get to a 100% success rate, then you only need to synthesize as many binders as you want, diversity of your designs. That'll make the DNA and the screening very cheap and make this an incredibly flexible technology. And software like Bindcraft, which has a significantly higher success rate than RFdiffusion, is kind of pointing in that direction. I think there's one of the challenges is there's kind of a limited pool of researchers currently which has the required skill set to kind of fully implement de novo protein design pipeline, because in order to select your target, select your model, and then create good parameters for the design, I think you need a really strong understanding of protein biochemistry and structural biology, and a really strong understanding of the dynamics and function of the system that you're targeting, but also skills and understanding of machine learning and Linux-based, Python-based computer systems are also quite important. And so that's important to some degree to do the design, but it's really essential for doing the development and the improvement of these tools. And ideally having a team which has, strong understanding of protein biochemistry, structural biology, and strong understanding of machine learning and computer systems that's well integrated is going to be required to get the best results from these tools. And that's basically something that we've developed and we're working to further develop in our laboratories. De novo antibodies are still difficult to generate. So antibodies are probably the easiest modality to rapidly translate into therapeutics because they're already being used as therapeutics, and we understand some of the rules about making them not immunogenic, and also the risk appetite for pharma and for commercial kind of development is smaller because they're more of a known quantity effectively. However, machine learning-based structure prediction still struggles to predict antibody antigen interfaces, although this is improving. So we submitted this target to CASP16, the structural modeling prediction competition last year, and this is a nanobody bound to hemoglobin. And basically, one out of, I think, the 40 groups that competed, one of the groups managed to accurately predict this structure. And this is, I think, the first time where in a competition, an antibody antigen complex has been accurately predicted. And this is something which, this is the target which both AlphaFold 3, Chai, and Boltz also failed at. So it suggests that there's continued technology development, which is going to make this prediction a lot more feasible. Another significant advance in this area is Chai 2, which was a program released by Chai Discovery a few weeks ago, which the code isn't available, sorry. So they just kind of did a press release showing that they've developed this technology, but they're claiming a 16% success rate for developing de novo antibodies against 50% of targets that they tried, which hadn't been targeted before, and like 68%, I think, for mini binders with a different scaffold. So it looks like technology is progressing, and whether or not these kind of company developed proprietary tools will become publicly available, I would expect that, academic open source researchers will catch up in time and keep moving the field forward. So I feel like we're sort of on a non-breaking wave of technology that's going to continue to push these approaches forward. Other challenges for translations into therapeutics include delivery and bioavailability. And so some reports, like the Cell paper on Th17 antagonists from the Baker lab, indicates that oral bioavailability for really stable de novo proteins might be possible. mRNA delivery is definitely going to be really relevant for some targets, especially like antivirals or things that have an intracellular target. And inhalation or injection will definitely work in some cases. And so scoping the best methods of delivering these tools in a way which is compatible with patient outcomes is going to be something for future work. And we still need to do more work to scope the stability, toxicity, and immunogenicity of binders. So there's really promising preliminary data showing that this might not be such a problem, but I think it'll be important to, fully profile how the human body reacts to these proteins and then incorporate that into our kind of design parameters so we can get the most out of this technology. Another really kind of cool example of how de novo design proteins might be used is this de novo design proteins neutralizing snake venom toxins paper from the Baker lab that came out in Nature a few months ago. And so basically they designed de novo binders to a number of snake venom toxins and showed that they could protect mice from the lethal effect of these toxins, with sometimes administered before and also administered after the toxin was administered. So this is kind of like a promising way that you could rapidly, do something in silico and translate it into something which could save lives. And also the mice that were treated with these de novo proteins didn't have any adverse effects from the treatment. So that's kind of a nice indication that there's good potential here. So that's it for my talk. I just really like to thank all the people that contributed to this work. So this is a partnership with Gavin Knott, Slab at Monash University, and Cynthia Taverneau is going to discuss some more work we've been doing using de novo design proteins in this forum in a couple of weeks. I'd also like to thank Maria Dramicinin from the WeHi Protein Production Facility, who was really instrumental in kind of the genesis of this project and also in the expression and characterization of the binders. I'd like to thank Twist Bioscience, who we've partnered with for DNA synthesis, and so they've been a really accommodating partner, both in providing us with DNA synthesis at a competitive price and also providing really rapid turnarounds so that we can, quickly translate our ideas into the wet lab and show that they work. This work was done at the University of Melbourne. Monash University was funded by a whole bunch of funding agencies that you can see here. And I'd really like just to thank the enthusiasm and contribution of the really talented people in my lab to this project, especially Daniel Fox, who's a PhD student who did the vast majority of the experimental work. So thanks very much and happy to take any questions.
I might get us started off with a question. Given what you know now, having gone through this whole journey, would you approach the same project differently?
I think no. I think it's hard to unpick what you've learned in the process, but this was such a great scoping and learning experience for us. And what I really like is it wasn't a super easy or model target. It was, we're basically targeting two quite flexible loops, and it wasn't the easiest target to design against. And it kind of gave us the result we wanted. So I think I would probably do pretty well, things mostly the same as I did them.
That's really interesting that you still want to have that mix of the traditional methods and the deep learning in there. And I think that's actually quite important to be able to validate the results in both directions. Okay, so turning to some other questions now. The first one I have here, I'm just going to read it out to you, is did you put any steps in place for increasing the chance that your binders were soluble and expressible, such as using the soluble MPNN model or using filtering metrics at the end of your workflow?
We just use the standard MPNN model. I think MPN, even the standard model, is quite robust at producing proteins that are quite stable and quite soluble. And so I think there would definitely be value depending on your application, trying different models or tweaking things. But this was our first go, so we just went with the default settings, and we still got a pretty good yield for most proteins at a 80 to 90% solubility rate. So it wasn't really a problem for us. Filtering metrics for solubility, I think, are still pretty tricky because you're basically, if a protein is not soluble, it's probably because it's not folding very well. And prediction programs still aren't amazing at addressing foldability. Even, I mean, your deep learning programs, they simulate how a protein folds, but they don't address how foldable it is. And so, in short, no, I guess we didn't.
Thanks, Reese. So there's a few different questions here, but people are quite keen to know how much it actually costs. So the first one is, can you give a ballpark price for the 96-well cloning from Twist?
Yeah. So I mean, I think it depends on your relationship with the DNA service provider. But if you're looking at, say, even $100 per, if $100 per plate is going to cost you like about 9,600, right? If you're doing 96, and if you can get that down to more like your $50 to $70, then you're going to be paying half that. So at the 96 scale, it is a significant investment. But I think that's offset by the fact if you have everything streamlined and working how well we've got it, the experiments that are required to validate your binders and to get something which has, a lot of potential aren't very expensive and are quite fast. So, if you're looking at the whole package of design, if you include, personnel hours, probably somewhere between, $15,000 and $25,000 to get your binders, that's still going to be cheaper than, an extensive nanobody campaign or something. And increasing success rates will just bring that down.
Great, thank you. Different topic again, there's lots of questions here, so I'm just going to pick a few. We probably won't get to everybody today, unfortunately. Did you find the sequences of the binders to be very unusual? Were they highly enriched in acidic and basic residues and with very few aromatics, for example?
Yeah, exactly. So default ProteinMPNN and the implementation that's used in that DL binder design package, it's kind of atypical, I guess. I mean, there must be some proteins like this. So you have a lot of surface charge. So you have a lot of glutamates and lysines on the surface. And yeah, you generally don't have a lot of tyrosine or tryptophan residues, which can make assessing concentration a little bit tricky. Bindcraft seems to have a more balanced kind of, the sequence you get from that is more balanced. But I think one of the reasons why the stability of these MPNN generated proteins is so high is that the really high performance of surface charge it forces the hydrophobic core by electrostatics, and so it makes the proteins quite stable. So it is unusual, but I don't know if it's necessarily a problem. I think downstream screening for applications will have to address that.
Thank you. Another question about the binders as well. Have you looked into the specificity of the successful ones?
Yeah. So, and this is some work that Gavin's lab has done on other projects that he can talk about later as well. But we took our successful binders and we screened them using BLI against 3 other Tom-dependent transporters and 1 other protein, and you get no binding response to any of those. So proteins with similar chemistry and a similar kind of overall fold. And I think it makes sense because the interactions that drive protein-protein interactions are quite specific. And if you've got proteins that are highly charged on the surface, they're not that likely to form an interaction with something. But yeah, we haven't detected any non-specific binding with any of our proteins.
I think you actually covered this in your talk, but I'm going to ask it anyway. You did do some structural characterization of your non-binders, actually, the question is.
No, sorry, go on.
Yeah, I think the question is more about, do you know why they didn't work?
No. There's a Simpsons quite, frankly, we don't want to know. No, we do want to know. But I think it might come down to the nuance that drives, affinity, and maybe these things do bind, but they don't bind well enough to have a phenotype. They don't bind well enough to outcompete the hemoglobin. And if that's the case, we didn't really see anything. And if you can see from that initial plate, there were quite a lot of binders that did appear to have some inhibitory activity, but it was relatively weak, and so we didn't go forward. And sort of colloquial stuff from talking to a few other people in the community who have been doing this, who have looked at lower affinity interactions with binders, found a lot more of them bound with maybe hundreds of micromolar affinity. And so I think it's still difficult to predict what interactions are going to drive your entropy and your enthalpy to get a high affinity interaction.
I guess that's where the biological part of the story comes in as well, that understanding how these things work within your biological system can help you understand why it doesn't work as well. It could be that just isn't biologically possible.
Exactly. Or there might be a conformational state you're not aware of that the protein occupies most of the time, and so the binding site isn't accessible. Yeah, there's all these ways to kind of fail, I guess.
Okay, I'm just scanning through. Give me a moment to read some of the other questions. There's a lot of excitement happening in the Q&A, and they're quite detailed questions. I just need a moment. Give me a second.
No worries.
Let me see what we have here. So we have a question that is, what proportion of your 96 designs had beta sheets? Did these express okay, or were the helical bundles more likely to be winners and grinners, or grinters that they have written?
There were some, we deliberately made sure there were some beta sheet designs in there. I didn't go over in detail which ones express, although because the proteins express quite well across the board, I assume, I think the beta sheets probably express comparably. But we didn't get any binders that were beta sheets. For it really is quite target dependent, I think. For certain targets, and the targets that Gavin's lab will talk about later, they ended up with beta sheet solutions, and they actually found they got better results if they used a model which biased towards beta sheet proteins. Against most targets, alpha helical bundles with RFdiffusion are the norm, but they're not always the best solution to the binder problem.
Thank you. Another question here is, can you elaborate, tell us a little bit more about the manual selection of candidates? Did you look for diversity? This person's wondering, there's lots of different approaches that can be applied, and what did you use?
Yeah. So struggle is maybe the wrong word, but this is something we've kind of mulled over, because it's like we don't have good ways of testing whether the metrics that we've chosen work or not, because there's a lot of things that could be a barrier to a binder not being a good binder. Diversity of fold is definitely something we would go for. And so you can rerun the sequence generation ProteinMPNN on the same scaffold, and so get the same scaffold with different sequences or changes in sequences. And so generally for our initial screening, we will try and go for stuff which has completely different folds. And then maximizing surface interface, maximizing hydrogen bonds and predicted delta G. And we haven't done, I think Gavin probably has, but we haven't done any like stability testing or in silico kind of predictions of that, but that might be able to help you as well. It seems to me that if you can get your in silico prediction metrics high enough, you've got enough of a chance of success that I'm not sure how much these additional selections sway it one way or another. It seems more target dependent than that detail dependent.
As I mentioned at the beginning, this webinar is part of a series, and the next one is on the 12th of August. We'll be joined by Dr. Cynthia Termino, who will be talking to us about AI-designed anti-CRISPRs. You can find out more about that webinar and the entire series on the Australian Biocommons website, along with other training events that we have coming up. You can also follow us on social media or subscribe to our monthly newsletter. If you are keen to connect with the Australian structural biology computing community and perhaps talk about some of these methods in more details, you can find out more about what's happening there, their monthly meetings and their Slack community on their website. And that link is also going to be shared into the chat as well. So thank you once again, Reese, for sharing your journey through this with us today. And thank you to everybody for joining us as well. As we leave, I'd like to acknowledge that Australian Biocommons is enabled by ANCRIS funding via Bioplatforms Australia. Thank you again to everyone for joining us. We hope that you have enjoyed this webinar and we look forward to seeing you in the next one. Until then, goodbye for now and enjoy the rest of your day.